So these are types of transfer that occur vertically. So that means, horizontally, not vertically. We already talked about mutations that are inheritable. They're passed on, stable changes that are passed on from generation to generation in the population of bacteria that are dividing and actively growing. But horizontal gene transfer can occur in organisms where they're not necessarily in a growing population. So first I'm going to talk about a process that was kind of accidentally discovered when a doctor in the late 1920s, Frederick Griffith, was studying a type of bacteria called Streptococcus pneumonia. So this is a type of gram-positive bacteria that's a potential pathogen, can cause respiratory diseases. And he was studying it actually using mice as a model. And he found that when he observed this type of bacteria under the microscope, that he found it in two forms. And actually, he could even see the difference when he looked at the growth on a petri dish. So different types of colonies, even though he knew it was the same species of bacteria. So he would start with the pure culture, and he would find some differences would appear. And that's smooth appearance or shiny appearance versus those that were not. And then when he looked under the microscope, he could, using negative staining, see that one had a capsule. So remember, what's the capsule? Where is it located? It's on the outside, the most outer, beyond the cell wall. It's glycocalyx, one of those sugary coats of usually polysaccharides. And it's associated with virulence. Okay, so one type appeared to have a capsule, whereas the others was not. And he wondered what the difference between these two bacteria were. And in a series of experiments looking at the response of mice to infection by these two different strains of the same species of bacteria, he was able to discover this importance of the capsule in the virulence of the organism. So in the first experiment, he knew from using mice as a model for disease that if he injected the bacteria into the mouse, it would get sick and die. Okay, so these were the original strain that he was working with. It was virulent. The mouse would die, and then he could sample from the mouse and grow bacteria that gave this shiny or smooth appearance on in the colony morphology of it on a petri dish, on a nutrient agar dish. Okay, so he found those. Then he also found that sometimes after growing it in lab, he found another type that didn't appear shiny. And he tested this to see if it would cause disease in mice. And using the same procedure, he injected the bacteria and found that no, the mice remained healthy and did not get sick and did not die. And he was able to culture from these mice some bacteria that had that same appearance as the original bacteria that he injected into the mice. So the colony morphology was not this shiny or smooth appearance. Then from our understanding of the control of microorganisms, lots of the studies from Louis Pasteur and others after him, he understood that he could kill the bacteria by using a brief treatment with heat, but they'd still be intact. So the heat killed bacteria. He injected into the an experimental mouse, and it remained healthy. So he proved that if he heat killed the virulent form, the form that usually would cause disease and kill the mouse, it was no longer able to cause disease. So in this way, again, he showed that it had to be live, reproducing bacteria in order to cause the disease. And then finally, he did a very interesting twist to the experiment. He combined both agents that didn't cause disease. So the unencapsulated live strain of bacteria, of Streptococcus, with the dead but virulent form of Streptococcus. And he got disease. And he was able to isolate colonies, bacteria that formed colonies that looked exactly like the virulent form of the Streptococcus. So this final step in his experiments implied there was some transfer of information between the bacteria. Sorry? Even though one was dead. Right? So how could a live unencapsulated bacterium gain virulence? What does it have to gain? Genetic material that encodes for what? In this case, capsule. Okay? So that's really the main difference between the virulent strain and the avirulent strain, is that the virulent strain has a set of genes that encode for this production of the capsule. And so now we know that the reason that Streptococcus heat-killed virulent Streptococcus when combined with a live avirulent Streptococcus, is able to cause disease is not because the dead bacteria suddenly became alive in some way, but because the dead bacteria, in the process of dying in this heat killing, they're broken apart. They release bits and pieces of their components within the cell, including bits and pieces of DNA. And these fragments of DNA that result from nucleases that are just in the environment that will chop the DNA into smaller pieces, these fragments are called naked DNA. And they're able to be taken up by a subset of cells that have certain competence. They're able they have receptors that are able to bind the DNA and bring it into the cell. So it must be that the avirulent form of the Streptococcus was a competent or in a proper state that it could receive this DNA, take it up. Once it's taken up, because they're the same species, there was a lot of homology. that was probably found, and if it wasn't immediately degraded, the DNA could then be, through homologous interactions and recombination, could be incorporated into the chromosome. And that's how the recipient DNA could then gain new traits, new attributes that would make it virulent. This is what we call DNA-mediated transformation.
DNA transformation involves the transfer of naked DNA into a recipient cell. In the first step, double-stranded donor DNA binds to specific receptors on the surface of a competent cell. One strand of the donor DNA is degraded by nucleases while the other strand enters the cell. The single-stranded donor DNA pairs with an homologous region on the recipient DNA and is integrated into the recipient genome by a breakage and reunion mechanism called homologous recombination. If there are any differences between the nucleotide sequences of the donor and recipient DNAs, the mismatch repair system comes into play. The repair system removes either the donor or the recipient strand and replaces it with the complementary sequence. Since either strand may be repaired, some cells contain the new donor DNA and others have the original DNA sequences. In the laboratory, cells are plated on selective media so that only the transformants will grow.
Okay, so remember, this is possible in bacteria because they don't have additional compartments which would prevent the entry of the DNA or the interaction of the DNA with the genome, the bacterial genomic DNA. So this combination, and also that it has this mismatch repair mechanism that may because it's naked DNA and it's still methylated. It's not like the new strand that's being synthesized that's not yet methylated. So it can't tell the difference between the parent strand and the correct strand, original strand, and the new strand, the recombined strand of DNA. So there is a chance that the new piece of DNA will be incorporated in the bacterium, in some of the bacteria, through this process of transformation. And it's not, in the case of this animation, it's showing you resistance that can be acquired. And this is something that is routinely done in the laboratory to manipulate traits that different strains of bacteria will have. But in the case of this experiment that Griffith did, it naturally happened by combining the dead virulent form of the bacteria with the live avirulent form. So it occurs in nature, and it also can occur, we now know the conditions to make cells competent by adding things like calcium chloride. That we'll be able to do it in a test tube in the lab as well. Okay, so DNA-mediated transformation is the take-up of DNA from dead or dying cells, okay? And it's a single strand that's taken up. And there are basically 4 steps. Entry of the single strand of DNA requires those receptors. Integration into the donor genome and mismatch repair that occurs such that the newly integrated DNA that's been integrated by homologous recombination is inherited by future generations. So while this is this is horizontal transfer of genetic information. There's no cell division required for this process to occur. Once it does occur, it will be passed on in horizontal transfer from generation to generation through binary fission. Now, there's another type of transfer that's called transduction. Transduction is different from transformation. It's still the gain of DNA. All of these horizontal gene transfer mechanisms are a transfer of DNA from one organism to another. But in the case of transduction, the vehicle which transfers the DNA is actually a virus that infects bacteria. So, and there's basically the mispackaging of the viral DNA that it accidentally picks up what in this cartoon here is a blue fragment of DNA, which is the host cell, the bacterial DNA, instead of the viral DNA, which is in red. And then next time it goes on to, it's in addition to its own viral DNA, so that still has the means of transferring to another bacteria or infecting another bacterial cell. But it can then pick up by accident genes that are bacterial genes. So the viral genes are significantly different from the bacterial chromosome, that there won't be any homologous recombination. But when you have a fragment of DNA from another bacterium being transferred into the new host bacterium, then there's a chance that, again, through homologous recombination, that fragment of DNA will be incorporated into the genome. In the same manner. And there are actually two types of transduction. It can just accidentally pick up a random piece of DNA, or there are areas that tend to be more commonly transferred because of homology or repeated sequences. And it may be plasmid DNA in the case of the specialized transduction. Now, the third type of transfer that I want you to understand is actually a direct contact between two bacteria. So before, it was kind of a random process where either a fragment of naked DNA would be picked up by a receptor on the bacterium, or a virus would transfer bacterial genes accidentally to the new host bacterium. But here there's a direct contact between two bacterial cells. And this requires a special set of genes by the donor cell. So only those cells that encode that specialized pilus called the sex pilus or the F factor will be able to form this connection to another cell. And only recipient cells that don't have these genes, only cells that don't have these genes will be able to act as recipient cells. So they must be F plus in order to donate DNA, and F minus in order to receive the DNA. In this case, we're not talking about the chromosomal DNA. We're talking about plasmid DNA, that extra chromosomal DNA that has additional features that can be advantageous. It can carry antibiotic resistance, for example. And because the plasmid, even though it's a very small fragment of DNA, it has its own origin of replication, so it can replicate independently from the chromosome. And so in that way, one strand of the plasmid can be transferred and then replicated in the recipient cell. So in the end, you get, once you add an F plus cell to an F minus population, eventually the entire population will gain this information and become F plus. There are some cases where in the case of high frequency recombination plasmids, where the plasmid DNA is actually incorporated into the recipient genome, into the chromosome. Okay, so let's take a moment to look at how this occurs.
Sometimes the F plasmid becomes integrated into the host cell genome. The host cell is now referred to as HFR, which stands for high frequency of recombination. The HFR is able to transfer some of the host genes to the recipient. The sex pillus contacts the recipient F- cell and pulls the cells together. The donor chromosome is transferred as single-stranded DNA starting at the origin of transfer. Gene 1, which is closest to the origin, is transferred first. Segments of the integrated plasmid are at the beginning and the end of the DNA being transferred. It is theoretically possible for the complete genome and the F plasmid to be transferred to the recipient cell. However, this does not happen because the donor and recipient cells will separate prior to the complete transfer of the donor chromosome. The transferred DNA becomes double-stranded. The donor DNA is integrated into the recipient cell's DNA by homologous recombination. The recipient now carries transferred genes, but remains F-, whereas the donor cell remains HFR.
Okay, so this is a little bit different than just the transfer of the plasmid, which would make the entire population. First of all, the entire plasmid is always transferred in completion. It's a much smaller fragment of DNA, so by the time the cells pull apart, the entire plasmid DNA is in the recipient cell. But in the case of the conjugation of the F plasmid, it doesn't transfer any of the chromosomal DNA. So there won't be any homologous recombination events occurring. Only in the case of the high frequency recombination is the potential for transfer of chromosome genes within the chromosome, so those essential genes from one bacterium to the next. So in addition to transfer by the F plasmid, there are other times when conjugation would occur, and that's in the case of transposition. So transposition can occur within a bacterium or between bacteria when it's combined with the process of conjugation. Transposition involves a mobile genetic element, not plasmids this time, but something called a transposon. And here's actually what is called a composite transposon. It actually has two transposon that are flanking a gene or a coding region. In this case, it's a gene encoding antibiotic resistance. But these transposons have mirror images, so they read the same way on opposite strands. T, C, G, A, T, G. Same thing here, A, G, C, T, A, C. And because they're palindromic or mirror images, they're very likely to recombine, very easily recombine with these with this homology that they have to each other. In between these sequences is a gene that encodes an enzyme that will cleave the transposon out of the DNA. So it's called transposase. So that helps these genes that might be flanked by these insertion sequences, helps them to be rapidly and frequently transferred from place to place. When they are transferred, they can disrupt, so they can recombine and disrupt a functional gene. So if it sits down in the middle of a coding sequence of another gene, the protein won't be, the mRNA won't be in the proper sequence to make a functional protein. And they can also, in the case of this type of composite transposons, transfer new genes to another bacterium in the process, during the process of conjugation. So for example, even between species, although it's rare, it can occur. So there was a case where two patients in the same hospital were studied, and that one patient had an infection with Enterococcus faecalis that had antibiotic resistance to the antibiotic vancomycin. Now, vancomycin is also used to treat Staphylococcus infections. And another patient in the hospital, while they were there, got infected with Staphylococcus aureus that was also vancomycin resistant. And they were able to isolate both Staphylococcus and Enterococcus from that patient. So what was proposed that happen is that the patient that originally came in without an infection was exposed to Staphylococcus that had acquired the gene for vancomycin resistance from the process of transposition and conjugation. So the gene for vancomycin resistance ended up in the plasmid that is unique to Staphylococcus. And that's because at one point there must have been both plasmids, both the plasmid from Enterococcus and the plasmid from the Staphylococcus in the same cell of Staphylococcus. And then after this recombination through transposition, it was inherited by each generation. So here's the step where transposition comes into play, where that homologous recombination. So there must have been an event where the genes jumped from the Enterococcus plasmid. Now, the reason that the plasmid for Enterococcus would not be passed on from generation to generation is that between species there are differences in the sequence of both the chromosomal DNA and the plasmid DNA. And so plasmids tend to be have certain differences between species. And these differences are in their sequence, in the reading of the DNA that code for sites that are recognized by restriction endonucleases. So enzymes that are able to recognize a specific sequence of about 6 base pairs and cut both strands of DNA. In that way, it would destroy the DNA. Now, the site for the restriction enzymes, these endonucleases that are found in Enterococcus would not recognize, there would be no sites that would recognize it in its own DNA because obviously it doesn't want to degrade its own DNA. But it would recognize Staphylococcus DNA or plasmid as having these sites. And it would digest the DNA. And the same thing happened with Staphylococcus has a set of enzymes, restriction enzymes, that it encodes in its genome. And those enzymes act on foreign DNA because they recognize a specific sequence called a restriction site. And they degrade that DNA. And this is thought to be a form of protection and also competition in between different species of bacteria. The reason that I'm going over this in a little more detail, and I'll talk about it again, is because restriction enzymes are an incredibly useful tool in genetic engineering. And so we're going to talk about genetic engineering in just a moment. But manipulating these bacteria in the lab has been made possible only with the discovery of this restriction endonuclease system. But before I go on to that, I just want to introduce one more type of mobile genetic element. We already talked about plasmids, which can be easily transferred through conjugation from one bacterium to another in the same species, or even different species, although they're at risk for degradation by these restriction endonucleases if it goes cross-species. Transposons, which are these elements because of their enzyme that they encode and also because of their palindromic sequences that are highly recombinatory elements that can move genes around from place to place within the same cell. And they can be transferred from cell to cell by the process of conjugation if they're part of a plasmid. And then genomic island, which is a larger fragment. So remember, plasmids are pretty small. They're anywhere from 1 to 10% of the size of the genome of a bacterium. Transposons usually carry a very small fragment of DNA as well, a single gene maybe, when they're these composite transposons. But genomic islands are larger fragments within the chromosome, or sometimes found in plasmids, that can transfer more than one gene at a time and give a growth advantage or some other advantage to survival or causing disease. So we talked briefly about Staphylococcus aureus, and we've mentioned in previous classes MRSA, methicillin-resistant Staphylococcus aureus. And I just want you to be aware that we'll actually be looking at Staphylococcus aureus by gram stain, which this is an example of this week. It's gram positive, and as you can see, it forms these little clusters. It's not motile. It doesn't form spores. It does have the versatility of being able to switch between respiration, anaerobic and aerobic respiration, and it's normally found as part of our flora on some people in their nose or on their skin or mucous membranes. Although it is a potential pathogen, so people who are carriers as their normal flora do have a greater chance of getting infections with this species. It causes skin infection, it causes respiratory infections like pneumonia. It does, in some cases, the virulent strains of bacteria can encode toxins that would be responsible for toxic shock syndrome, so staphylococcal toxic shock toxin. And it can also be the cause of food poisoning. And this is toxin mediated as well. But the difference between a strain of Staphylococcus aureus versus a strain that's MRSA, methicillin-resistant Staphylococcus aureus, is the presence of this R plasmid. R plasmids are a type of plasmid that carry resistance to drugs or even disinfectants. So here you can see that this is a single plasmid that can be transferred in its entirety through the process of conjugation from one bacterium to the next. It's not just giving antibiotic resistance to a single antibiotic or a single disinfectant. You're getting a variety, 4 different resistances plus disinfectant resistant gene in one fell swoop. So this is why our plasmids and resistance can rapidly spread in a population if the pressure is there for these certain strains to persist. So in the presence of an area where antibiotic is being used, the spread would be facilitated, the spread of these plasmids, and the survival of these strains would be facilitated as well, because those that didn't carry the resistant genes would be killed by the antibiotic. Okay, are there any questions before I go on to talk about biotechnology? Okay, so from Chapter 9, I hope that you will understand what recombinant DNA is and how it differs from chromosomal and plasmid DNA. 3 ways that biotechnology uses microorganisms or members of the microbial world. 3 different proteins that are produced by genetic engineering, or 3 types of benefits that we can gain. And how restriction enzymes work and are essential to genetic engineering. What a vector is, how it's used in genetic engineering to carry fragments of DNA that we're interested in studying or transferring, or for use in production of proteins. And I'm going to talk specifically about the cloning and production of the human insulin gene. So biotechnology, your book gives a definition of using microbes or microorganisms to produce products. But really, it's to produce products that help to solve problems or have some benefit. So it's the use of things like bacteria, yeast, viral DNA, in order to create a combination of DNA. Some of them are used as carriers. Some of these mobile genetic elements, like plasmids, are used and manipulated in the lab, and then the organism, such as a yeast or a bacterium, are used as vehicles for producing protein from these genes that we've manipulated. So recombinant DNA is a combination of DNA from two different organisms. Originally, in I think it was 1972, the first recombinant DNA molecule was created by combining two viruses, a monkey virus and a bacteria virus. Then later, just a matter of 3 or 4 years later, the first transgenic organism was made. So there's manipulation of the molecules to form recombinant DNA. So that's DNA from two different species. And the recombinant DNA isn't always very useful in its form as DNA, purified DNA. Often it has to have an organism so that you can produce proteins from those genes that you've recombined. So that once you've put this unnatural or foreign DNA into an organism, it's called a transgenic. Okay, and the first transgenic organism was bacteria. That was E. coli bacteria that had been given a gene on a plasmid that gave it resistance to a certain antibiotic. So when we're talking about organisms, they're called transgenic once they have this newly created DNA. And the process of genetic engineering is manipulating using these biochemical techniques to produce a product or solve a problem. So the tools that are necessary in order to create recombinant DNA are these restriction enzymes. And it's thought that they are a defense system, as I mentioned. They're naturally occurring in bacteria. Each bacteria genus will have a different restriction modification system. Not all bacteria have this system. But basically, it's a combination of an enzyme that cleaves DNA, both strands of DNA, so a double-stranded cleavage called an endonuclease, and a methylase, which adds methyl groups to the DNA. So the methylase would be protecting its own DNA from the action of the endonuclease. And the endonucleases or restriction enzymes can be thought of as molecular scissors because there are many different types. For example, this one that's shown in the diagram here, EchoR1, only recognizes the GAA-TTC sequence. Okay, so that's the only sequence it recognizes. It won't cut anywhere else. If it has GGC-TGA, it doesn't cut there. It won't sit down and recognize that site. So if the host bacterium doesn't have any of these sites, then it's essentially protected from digestion by these enzymes. But foreign DNA, any DNA that's introduced into the cell that would have these sites, could be degraded through digestion, through these double-stranded breaks. Now, some restriction enzymes are able to just cut clean through and leave blunt ends, whereas others leave what are called sticky ends or cohesive ends. So they leave an overhang of single-stranded DNA. This is the type of cut that's useful for genetic engineering and creating recombinant DNA. That's because by cutting both the plasmid that you're using in the creation of the recombinant DNA, as well as your DNA of interest. So let's say you want to create a recombinant molecule of yeast genes in a bacterial plasmid. So this might be used to study what the function of the genes are in the yeast. And so that the entire genome of the yeast would be digested by this restriction endonuclease, so that fragments of various sizes would all have these ends that could then be annealed or attached in a test tube. So the fragments that are generated are then combined with the gene from a foreign organism that's also been digested with these same restriction enzymes and the enzyme DNA ligase in order to get this covalent link. Now, of course, we can't really manipulate everything so precisely that we know which plasmids are simply reconnecting with their own plasmid DNA and which ones are getting the foreign DNA. So we have to have a way of differentiating that after we do the creation of the recombinant DNA. So we won't always get recombinant DNA. It may still be just the regular plasmid DNA that results from this process. But when you do get a successful introduction of the foreign DNA into the plasmid, then that's a recombinant DNA molecule. And in order to be useful for biotechnology, plasmids that are used as vectors or cloning vectors to create recombinant DNA need to have certain features. So we don't just use any plasmid. We use plasmids that, first of all, they have restriction sites. So they have a site where we can insert our DNA and create a recombinant DNA molecule. But then we need to be able to tell the difference between just the plasmid that religated and it's still its original non-recombinant DNA, it's plasmid DNA, versus those that have recombined. So you need a selectable marker. And usually it's where there's a marker that would be disrupted by the presence of this foreign DNA. So it's some trait that can be measured or visualized that will suddenly disappear once you've got your recombinant DNA. So these features of a successful cloning vector, whether it's a bacterial plasmid or a yeast chromosome or a plant plasmid, or even a virus, such as a bacterial virus called bacteriophage. All of these can function as vectors because they can have their own origin of replication, so that's required. You need to be able to copy the DNA that you're interested in. It needs to be copied once it's put into the host organism to create a transgenic organism. If it doesn't have the origin of replication, that DNA won't be copied and it'll be useless. It needs to have a marker, usually antibiotic resistance, so then you can know which organisms became transgenic and which didn't. So which bacteria have the plasmid in it. Not all are going to take up the plasmid in this process. And then it has a second marker to tell which of the bacterial hosts, for example, have the recombinant DNA in it, not just the plasmid DNA. So let's go over that. So once you've got that cloned recombinant DNA, okay, so you have your DNA of interest, your plasmid, now you have recombinant DNA, now you introduce it into a population of bacteria, some of which will take it up. Once you have that, then you have your transgenic organism. And that's the basic steps in creating a transgenic organism or genetically engineering an organism such as a bacteria. There are other factors that are important, such as if you have a plasmid that makes a lot of copies. So remember, plasmids don't just have one copy in a cell, they can have many copies. The more copies you have, the more expression of that gene you're going to have. And really, most of the time, what we're interested in getting is a protein product from that gene of interest. But it can be used for studying entire genomes or just for inserting a specific gene, as in the case of the production of human insulin. And it can be used for protein production, DNA production, research or our understanding of how genes are regulated, or what the function of certain genes are. Also, things like vaccine production and the production of medically relevant proteins like insulin. So I'm just going to tell you a little bit about the story of insulin, but you'll find, if you look carefully, you can find even in the supermarket genetic engineering products in your food. So things like cheese now traditionally was made with animal rennet, so enzymes isolated from the gut of pigs, for example, was necessary for the production of cheese. Now, either yeast or bacterial enzymes are often used more cheaply and more efficiently produced. And similarly with pharmaceutical products, in the case of diabetes treatment, so remember type 1, the early onset, is a lack of insulin, the hormone. So how does that work? What's the problem with people who have diabetes? What's their dysfunction? Yeah, one or the other. So in the case of the type 1, they don't produce insulin. And so what happens when they eat a typical meal? Right, so their blood sugar levels are not properly regulated. And they either get very high levels where they feel like very thirsty or hungry or tired, or very low levels where they get very weak and faint and can have headaches. But the traditional treatment for this was insulin shots. And the insulin was isolated and purified in a very laborious way from animal sources. So where is the insulin made in our bodies? In the pancreas. So things like cow or pig pancreas was harvested from pigs, then homogenized, and then the protein was purified from that source. And it was a very laborious, expensive way of making insulin, and it had also the drawback that there were sometimes allergic reactions that would occur in patients because of the similarities between the animal and the insulin proteins. So shortly after the first creation of recombinant DNA and the first creation of a transgenic organism in bacteria, Scientists at a company called Genentech, the first biotechnology company, they thought of a way that they might be able to produce insulin, human insulin, rather than animal insulin, which wouldn't create these problems of interactions of allergic reactions in bacteria, in a transgenic organism. And it's really by using the same methods and tools that were used in the creation of recombinant DNA in transgenic organisms. So they used restriction enzymes. They used a bacterial plasmid as a vector. They were able, because the human gene for insulin had been discovered, so the entire sequence was known at that point. They were able to make what is called complementary DNA. cDNA is basically the gene without the introns, so it's kind of similar to mRNA, but it's the nucleotides that would be in a sequence of DNA. So they were able to synthesize it in the lab because it's a fairly short gene. They actually had to synthesize two genes, the two fragments A and B that make up the human insulin gene. So they had to do two processes of creating recombinant DNA. But then once they've got their recombinant plasmid, they used an artificial means of introducing it through transfection, introducing it into a host cell. E. coli bacteria was used to create this transgenic organism that had the genes that encoded for human insulin, and every time that the plasmid DNA was replicated, or every time that it was expressed and translated into protein, then that protein would be released from the bacterium. So basically, by growing up a lot of these bacteria, these transgenic bacteria, they were able to have a lot of insulin be produced in the media that they were growing these bacteria in. And so they could just spin out all these bacteria by centrifugation. They could separate the liquid portion of the media from the solid portion, the cells. Take that liquid portion and purify human insulin, the A portion and the B portion. And then at first, just through random combination, they were able to get the proper interaction of human insulin A and B to make a mature insulin, functional insulin molecule. That wasn't very efficient, but now they've tweaked the system so that it's much more efficient in producing functional insulin. But this was the first demonstration that a human gene could be produced through this process of genetic engineering. And now it's the way that all insulin is made. So let's go over the process of how they actually did this before I go over the benefits. So the first step was to chemically synthesize the DNA. Okay, so they knew the sequence of the human insulin. Okay, but remember, in human DNA, we are eukaryotes. We don't just have the coding region, we have exons and introns, right? So we have these sequences, parts of the sequences that would not be part of the messenger RNA, and so would not be part of the protein. So you have to get rid of those. So in the process of making cDNA, it's actually taking using enzymes to splice out the introns and taking just the exons, just the coding region of the gene, and this is cDNA. It's actually reverse transcribed from mRNA. So remember the mRNA, once it's produced, it's spliced, so it just has the exons. So we call this mature mRNA. And then it's in the test tube, it's transcribed back into DNA. So remember, the nucleic acids in RNA would be different from DNA. But in this process, you're kind of doing backwards or reverse engineering of something that would never happen in an eukaryotic cell naturally, because we don't have this enzyme called reverse transcriptase. But some viruses do, and so using this viral enzyme, we're able to make complementary DNA. Or it can be synthesized in the sequence can be directly synthesized in the lab. So once you have that fragment, it has to be cut with your restriction enzyme of choice. So here in this case, it was EcoR1. The GGA-TTC sequence, both the cDNA and the plasmid vector. Okay, the vector is just the carrier for this foreign DNA. In this case, it was a bacterial plasmid from E. coli. Also cut with EcoR1. Then you recombine it. And you get three things. You get the plasmid alone. You get the recombinant DNA. So that's the plasmid plus in this case, the insulin DNA. Okay. And then, so you get two products. So then when you introduce it into the host, in this case, the host was E. coli. Some of the host will not get any plasmid. Or recombinant DNA. Some will get the plasmid, and some will get the recombinant DNA. So now, how do we figure out which one is which? So you have to do a selection. One selection that will be done immediately is after you've done the transfection, you grow the bacteria on Okay, 5. Okay, so the first thing you do during the selection is grow on, for example, streptomycin. That's an antibiotic that will kill all of the bacteria that don't have our plasmid vector. Okay, so the plasmid vector has these traits of the origin of replication, two selectable markers, one with antibiotic resistance and the other in that site where it will be disrupted if you get recombinant DNA. Okay, so remember those 3 features of a good cloning vector. So grow on streptomycin, so bacteria without plasmid or recombinant DNA will die. Okay. Second thing is to either replicate plate or grow on, and this can be done in one step. It can have both streptomycin and a selectable substrate like XGAL. This is a modified form of galactose. Now, the second selectable marker is in a site that will encode for beta-galactosidase, which will use XGAL as a substrate and produce color to give the colony color. So you can actually just look at the plate. I think I have a picture of this, yes. So you can just look at the plate and you can see that in the case of the bacteria that just have the plasmid, so they're transgenic organisms because they have the plasmid in them, or no, they're transformed because they have the plasmid, but they're not transgenic because they don't have that extra foreign DNA in them. So with the plasmid alone, you'll get blue colonies because they'll have the functional enzyme, and it will use XGAL as a substrate. But here, in the case of the transgenic bacteria, those that took up the plasmid that had the foreign DNA in it, the insulin gene, for example, those will be white. So right away, you know you can then select a white colony just by using your inoculating loop to pick up just that one colony, put it in fresh media, grow it up. You have a pure population of your transgenic organism. And what they do now in biotechnology companies like Genzyme, they have that big building on when you drive along Storo Drive. They have that big brick building. It has enormous vats where you have thousands of liters of media that will support the growth of bacteria. And that's how they purify the drugs that they produce from these transgenic organisms. So the advantage in the case of insulin is it's cheaper. The bacteria grow a lot faster than a pig or a cow. You don't have the risk of any allergic reactions. It's animal insulin versus human insulin, so it's safer also. Does anyone have any questions about that? I went over it pretty quickly. I'd like you to understand that process of cloning, like the importance of the example of how we've used microbes, in particular bacteria, in genetically engineering and producing useful products. Now, it's not just restricted to bacteria. Yeast may be necessary in the case of proteins that need modification. So remember, bacteria don't modify their proteins after they're produced. But in eukaryotes, we often have these post-translational modifications where they're cleaved into their active form, like there's the pre-enzyme or the pro-enzyme and the active enzyme, things like that. So the bacteria might not be able to produce a functional protein in all cases. You may need a eukaryotic organism to be your transgenic organism. And so yeast can be also very important for serving as host organisms. Also, you may need to be able to clone a larger fragment of DNA, in which case the yeast chromosome could serve as a useful vector. Plants are, so animals are not the only genetically engineered organisms. Plants are commonly genetically engineered. So we have BT corn, which is genetically engineered corn. Also, there's genetically engineered cotton and potatoes. All of these changes that have been introduced either give the plant some advantage, like it's resistant to common pests, or they're made resistant to herbicides, so they can grow better, but without having the problem of other plants growing in the crops. Also, they can add nutrient values. So rice has been genetically engineered to include vitamin A, which improves the health of those individuals that don't get as much meat in their diets. It's an important nutrient that can improve the general population's health. And plants have a special type of plasmid, so you can't use a bacterial plasmid in a plant, although there is a small circular fragment of DNA called the T plasmid that comes from a type of bacteria that causes tumors in plants. So not all organisms, so for example, an E. coli plasmid vector would not be functional in plant genetic engineering. But your book goes over this process of using the T plasmid and how it's used in genetically engineering plants in sufficient detail. So that's just another example of a transgenic organism. It doesn't have to be a bacteria or yeast. It can be a plant. That's the transgenic organism.